Hello and welcome to this week's mini video on point mutations. To help us master this material, we're going to use the Jacobs stick figure family. Karen, the mother, was very interested in learning more about her children's genetic mutations from her own DNA sequence, so she hired the NCSU Genetics 311 class to investigate. Let's get to work. First, let's look at Karen's genetic composition. We were given those two DNA sequences. What's the problem with the bottom strand? It isn't in the 5' to 3' direction. Let's flip it and move on. Next, we transcribed her DNA into mRNA by switching all the bases tyrosine to uracil. Now that we've done that step, let's identify the start and stop codons. The start codon will always code for the amino acid methionine, which is AUG. The stop codon will have the code UAA, UAG, or UGA. To compare Karen's mRNA sequence to her children, we'll be using the sequence that results in a 4 amino acid protein. Therefore, we must use the bottom strand because it codes for a 4 amino acid protein, whereas the top strand codes for a 3 amino acid protein. With this in mind, I translated the mRNA sequence using the chart to take a better look at the amino acid composition. We'll need this later to compare Karen's mRNA sequence to her children. Before we begin this investigation, we need to look at the different point mutations that could occur. Point mutations occur in the substitution of one base for another. In transition point mutations, purines are substituted for a different purine, and pyrimidines are substituted for a different pyrimidine. For example, adenine can be substituted to guanine and vice versa. In transversion point mutations, purines are substituted for a pyrimidine, and pyrimidines are substituted for a purine. For example, guanine can be substituted to cytosine and vice versa. Finally, let's look at the effects of these point mutations at the protein level. Missense mutations completely change the amino acid and will thus alter its functionality. Nonsense mutations will change the amino acid to a stop codon and will result in a shorter protein. Silent mutations changes a codon but codes for the same amino acid. And lastly, read-through mutations changes a stop codon to a codon that codes for an amino acid and will result in a longer protein. Now let's investigate the point mutations that take place in the Jacobs family. Karen has an older son named Tucker. His mRNA sequence is given and compared to his mother's mRNA sequence. Indicated in red, uracil is substituted for cytosine, changing the amino acid valine to alanine. With this in mind, what kind of mutation occurs? A transition point mutation that is missense. Now let's move on to Camden, Karen's oldest daughter. Also indicated in red, cytosine is substituted for uracil, but the amino acid doesn't change. This is a transition point mutation that is characterized as silent. Now, looking at Jake's mRNA sequence, we see that adenine is substituted for cytosine and the stop codon is changed to the amino acid tyrosine. This is a transversion point mutation that is read-through. Last but not least, Alice has the mRNA sequence that changes adenine to uracil and lysine to a stop codon. This is a transversion mutation that is nonsense. Here's a summary of the mutations that we discussed, and here are my resources. Thank you.